All right, so we're going to pick up our discussion with meiosis, specifically with the processes of spermatogenesis and oogenesis. Meiosis is going to be a part of the formation of sperm and egg cells. Following meiosis, the daughter cells are going to then mature to become fully formed gametes. So spermatogenesis is a continual process in the testes starting around puberty. About 300,000 sperm cells are going to be made per minute. Over 400 million sperm cells can be made in a single day. The primary spermatocyte, so that's going to be this cell right up here, is initially a diploid cell, meaning that it's going to have, well, here, let me pause. What does a diploid cell mean? How many chromosomes are going to be present in this initial cell? So the primary spermatocytes are going to be diploid. They're going to divide during meiosis 1 to form two secondary spermatocytes, which are going to be, at this stage, are these cells going to be haploid or diploid at the point where we have completed meiosis 1. Secondary spermatocytes are then going to divide during meiosis 2 to form four spermatotids. The chromosome in the secondary spermatocytes is going to be duplicated and consist of two chromatids, whereas those in the spermatids consist of only one. So here we had two sister chromatids, now we have only daughter chromosomes. We end up with four haploid cells. Now I want to ask you, are these haploid cells genetically unique or genetically identical to the original primary spermatocyte? Yay, I don't know. I'm going to give you a chance to answer. The spermatotids are then going to, once they have divided out, they are then going to mature into spermatozoa and eventually become sperm cells, with the fully formed flagella, acrosome around the head of the cell, and then the mid piece portion. In the case of spermatogenesis, all four daughter cells will eventually become sperm cells. The reason we say this is because the process is a little bit different in oogenesis. In the case of oogenesis, we will form four haploid daughter cells. However, not all four of them will become eggs. Ovaries contain many immature follicles. Those immature follicles contain a primary oocyte, that would be this region right here. That's going to be arrested in prophase 1. So it's going to undergo an initial division during meiosis 1, where it's going to actually produce a polar body and a secondary oocyte. The secondary oocyte is going to receive almost all of the cytoplasm, all of the organelles, all of the nutrients. This is also going to then begin meiosis 2, but it stops at metaphase 2. It's not going to complete its total division unless it's fertilized by a cell. Now, those polar bodies that are off to the side, those polar bodies are ultimately going to just hold the discarded chromosome. The secondary oocyte is going to leave the ovary during ovulation. It's going to enter the uterine tube. That'll be this secondary oocyte is going to move into the uterine tube. And that's where it's going to be either fertilized and complete its meiotic division, or it's going to be unfertilized and then sloughed off during menses. Towards the end of meiosis 2, there's going to be one egg and either one or two, possibly three different polar bodies. Those polar bodies are going to disintegrate, which is a way to discard the unnecessary chromosomes while keeping most of the cytoplasm in the egg. So one thing I want to give you a moment to just think about: why do we form these polar bodies? If we start off with one primary oocyte and we end up with one egg, and that one egg contains almost all of the nutrients, cytoplasm, and organelles, why do we undergo this process to begin with? What is the point? If we're not going to be producing four eggs, why do we undergo meiosis? So in other words, why do we form these polar bodies? Another way to look at the question is, why do we form these polar bodies? What is the point of these polar bodies if they're ultimately not going to become eggs and they're ultimately going to be discarded through the process? All right, so I'm going to leave that out there for you. Please feel free to ask, and I'll be happy to discuss it in our evening meetings. But that is one question that I want you to just sort of mull over and think about. What is the point of producing those polar bodies? All right, So I just want to give you just a quick moment. We're going to just summarize the point of meiosis. We're going to spend a lot of time in our next section comparing meiosis 1 with meiosis 2. We're going to compare meiosis with mitosis, and we're going to look at all of the different stages. But I just want to summarize with a quick significance of meiosis is that its primary function is going to be to keep the chromosome number constant from one generation to the next. We're going to ensure that each generation has a complete diploid set of 46 chromosomes, no more, no less. Now, the interesting thing about meiosis is that it also results in genetic recombination. Genetic recombination ensures that the offspring are going to be genetically different from each other and from their parents. This is how we introduce genetic variation into a population. This is going to result from crossing over events, independent alignment, independent assortment of those chromosomes, and then also fertilization. Fertilization also introduces a huge element of genetic variability. This is going to generate the diversity that is needed to survive in changing conditions. We're going to see that mitosis occurs, or asexual reproduction occurs a lot in bacteria. Bacteria don't undergo genetic recombination. Instead, what they do is going to clone themselves for each subsequent generation. They're going to simply make a copy of themselves and duplicate. Now, this is a really useful strategy if your environment never changes. If you can assume that your environment is going to be constant and there's never going to be a change, you might as well just make a bunch of clones of yourself and assume that you're optimized for that environment. The problem is that environments change, and with environmental change, we're going to see natural selection. This will be a huge point in a later chapter. And with natural selection, it's very important to have genetic variation in a population. So again, this isn't really something that I want you to focus on right now, but it's going to come up later on, so I want to make sure that you've at least heard it before. But genetic variation is going to be very important in changing environments. All right, so for this section, you should be able to explain how, following meiosis, the chromosome number of the daughter cells compares to the chromosome number of the parent cells, explain how meiosis reduces the likelihood that gametes will have the same combination of chromosome and genes, summarize the events that occur during the two cell divisions of meiosis, and then compare and contrast the stages of oogenesis and spermatogenesis. Also, you should be able to compare and contrast the outcomes in terms of the number and genetic variability of the daughter cells produced by oogenesis and spermatogenesis.